using this ruby aperture in the hydroshear, passing high pressure water through, we're going to tear our DNA fragments into smaller usable pieces. With those pieces, we'll run a gel and excise the band using our ladder that is giving us a fragment that is more useful. So that usually ranges from 300 to 600 base pairs. So you'll have this hydroshear shed up. You'll run your sample through, collect the sample, run the gel, excise the band, melt that band, put that into a solution of LB broth and bliss, grow that up overnight at 30 degrees, basically giving the E. coli a chance to take up our fragment and to multiply. Not everything is running in the lab, so it's going to be a little bit difficult to explain everything because you won't be able to see it, but I'll try to do my best. So when we have the transformation taking place in E. coli, it's taking up our recombinant DNA, we hope. And we transform that using an electroporator that used to be in the next bay, basically creating a whole. E. coli takes in the fragment, it grows overnight. And our three possible outcomes for transformation is that we either take an E. coli cell that closed on itself without taking an insert, we took up an insert but it didn't contain our recombinant DNA, or what we're hoping for, that it did take up the recombinant piece of DNA in the E. coli cell. And so that gets passed through. Any questions so far? I'm sorry? Yeah. Well, in 454, in the new technologies, we use a liquid nitrogen. Yeah. Oh. I like that. So we grew our E. coli cells overnight in our broth, and what we do now, this isn't a real example, so we spread them on these agarose plates, just like an agarose gel. Using sterile beads, we'll put our LB glyphs, whatever, different libraries, different species, obviously no cross-contamination. Put the sterile beads on there and basically shake. And so when I first started working here, an operator in the afternoon shift would probably hold 6 of these at a time and go like this about 600 times in a day, and have to do that like 42 times per batch at 6 batches. So your wrist would probably hurt. So the cup that was shown on the outside spawned this idea. An operator took a napkin one day and said, why don't we just come up with something that can hold the plates for us, and we'll just put the beads in manually and then just move it around this way, so we won't have to hold the 6 at a time. This was submitted into the Ergo Cup in 2007 in Dallas, Texas, I believe, and we won the Ergo Cup for that award on a team-driven idea. Basically, an operator just had an idea, submitted it to the engineers on site. Those engineers took it to the engineers at LBL, and they fabricated this piece of machinery. Later, that actually gave rise to this. So now, you actually put this on here, and a program, which obviously isn't open, you can select, and it's called the Miranda file, I think it is. So Miranda is an operator that basically recorded all of the steps that she was doing with her hands on this machine, transcribed it to this machine. They recorded it, you just press play now, you don't even have to do anything, and then it just does the same. So it actually eliminates any kind of disparity between how many plates an operator may handle, how many times he or she may rotate, how many beads they may use, etc. The end result of spreading your transformation stock on this plate is that you're spreading evenly around, we hope, the E. coli cells with our recombinant DNA. These are grown up overnight. We'll have growth of what we hope to be are the white colonies that we'll select for in the QPix room. You'll have satellite blue colonies that didn't take up our DNA at all, but we'll select against those using the QPix machines. Any questions so far?
This is basically a spread plating machine.
Yes.
I see.
questions, and we'll continue.
The bead acts like a spreader.
Okay.
That's the way that I understand it.
We have this bioassay. This is a pre-picked bioassay, and if you'll notice, this came out with growth. Those colonies are a little small, but the computer has software on the QPix that'll determine whether or not we should pick them. So what's happening here in this is the QPix room. This is where the colony picking takes place. These QPixes were actually designed by an engineer on site. He actually walked through our group in the lab code. Anyways, that idea came up around 12, 13 years ago. We actually gave the idea to a British company, Genetics. Now they make it for us. Take home message here. You take one of those bioassays, you put them on this stage here. This CCD camera located on the QPix head scans the entire bioassay, and it tells the software, hey, this is how many colonies that we have here. These are what we determined to be too small. Those parameters we usually set, saying that in this range, these are DNA, these are E. coli cells that we can transport into our finished destination plate, which usually sits here. I don't know if there are any. I'll grab one if there are. As you can see. So, what once was those large bioassays that sit here, the destination plate sits here. You scan your image, you set your parameters on the software. The colony picker then sends this colony head, fires a pin directly, one pin, 96 times. So the pin fires. You got it? Okay. So then it fires 96 times, comes over to this plate, puts it in, cleans itself in ethanol, dries itself using light. It used to use air in these older models. These newer models, more ergonomically correct, they have a stacker system. You just load these, it automatically takes a plate. You notice that the newer models also have 2 stages where you can put bioassays. Whereas for these older ones, excuse me, you put these on by hand. A lot of wrist injuries doing it like this. Also, in my tenure here, there wasn't height adjustable tables. Now there are. Anyways, 96 wells, 4 times 96, 384. So now you have a 384 well destination plate. 1 unique colony per well. This just contains LB glyphs. We can freeze these. That's why we need it. Any questions from here?
So when you have these multiple do you have to make duplicate plates from each colony, or do you have just one? You know, the colonies pick once. Does it get inoculated into duplicate plates?
No, just one. Once the pin is fired into that colony, that's it. It'll just go in and inoculate into one quadrant of the plate, and that's it.
Is that liquid? That's liquid broth, right?
Yes. That's actually dispensed on that machine there. It has an automatic plate crane arm that'll handle the plates. The operator will just load those stacks individually, dispensing glyphs, priming the glyphs on the right side. That robotic arm will just pick up a plate, remove the lid for you, dispense the plate, put the lid back, and then stack it for you. Again, 4 years ago when I started, we didn't have the arms, so you just had to load those before. Even prior to that, they didn't have the QPix machines. They used grad students with toothpicks and just kind of one colony at a time. So we've come a long way.
So the clock basically fires 4 times per plate. Does the plate